Okay, so we're going to take a look at the anatomy of the central nervous system. We're going to look at it from a functional point of view as opposed to a hard anatomical point of view, mainly because it's very hard to delineate where one structure happens to be versus another structure within something that is a gelatinous mess like we talked about before. So we're going to start with looking at the cerebral cortex and the lobes of the cerebrum. So what we have to remember here is we have somatic organization that's based on what's referred to as contralateral hemispherical organization. What this means is that the left side of the cerebral cortex is going to control the right side of the body, and the right side of the cerebral cortex is going to control the left side of the body. The control and coordination of everything is made through connections through the corpus callosum. Any misfiring within the corpus callosum leads to issues of epilepsy. In particular, what we usually refer to as grand mal and petit mal epilepsy. What they can do is they can go ahead and surgically lyse the corpus callosum. The problem with lysing the corpus callosum is that it leads to what's referred to as hemispherical neglect because there's no cross communication. What the corpus callosum is going to allow to take place, and we'll look at this when we look at the midbrain structures here, is that it allows for communication from the right hemisphere to the left hemisphere so that both hemispheres know what's going on. There is additional regionalization and specialization based off of motor control and perception of sensations within the cerebral cortex. The one thing we have to remember is that even though we talk about lateralization, there's no such thing as brain specificity. There's no right brain specific person, and there's no left brain specific person. There's just a brained person. There's a lot of misconceptions about, oh, if I have this skill, I must be left brain. If I have this skill, I must be right brain. We do see a higher density of neurons associated with specific skills in specific regions, but you're going to be using both brains at the same time. Additionally, you are using 100% of your brain at all times, regardless of what people might say. The cerebral cortex is just like any other tissue. If we don't use that tissue, we're going to lose that tissue, which means if we only use 10% of the cerebral cortex, we're only going to maintain and keep 10% of the cerebral cortex. Within the cerebral cortex, we have three distinct divisions that we talked about before. We have the hindbrain, the midbrain, and the forebrain. Evolutionarily speaking, the hindbrain is the most conserved. That means that we see it across all vertebrae. The midbrain is the next most conserved. The forebrain is the most derived. That means it's the most evolved. Now, having said that, there's no such thing as a reptilian brain or a bird brain or a amphibian brain within human populations. It's a misconception based off of the evolutionary conservation of some of these structures. Within all mammals, there is the conservation of the forebrain across all mammal species. Along with the forebrain being the most evolved, it's also the most derived, which means that it's going to go ahead and adapt and have plastic changes with aging and with development, which can become problematic, particularly for head trauma victims, as head trauma victims, particularly young aged head trauma victims, can have a retrograde issue in terms of the derivations due to trauma not allowing full development of structures within the frontal lobe, in particular the prefrontal cortex. So we're going to start looking at the cerebral cortex, looking at one of the five brains. We'll look here at the hindbrain. The hindbrain is going to be made up of the structures of the cerebellum, the pons, the medulla oblongata, and what we refer to as the brainstem. The brainstem is the junction point between the medulla oblongata and the spinal cord itself. Within these structures, we have some anatomical structures that we have to worry about in terms of flow of cerebral cortical fluids, which we'll talk about in another lecture. So here is the hindbrain in a cross-section, as well as looking at a superior view of the cerebellum. So notice that within the hindbrain, we basically start at the midbrain structures, the tectal plate, where you see the pineal gland. We also have right where you see the pineal gland, we'll have the collicules. And then we have what's referred to as the mammillary body, which is the most superior edge of all of this. At which point in time we get to the mesencephalon. That is the remnants of the mesencephalon that developed during fetal development. Everything that's going to be inferior to those tectal plate and mesencephalon is going to be hindbrain. So you notice there's a cerebral aqueduct that's going to be connecting with the ventricles within the cerebral cortex, allowing for cerebral spinal fluid to flow down into the spinal cord. The aqueduct is going to lead anterior to the tectal plate, but posterior to the mesencephalon. It's going to continue through anterior to the cerebellum, leading to the fourth ventricle. It will then exit out through what's referred to as the central canal of the spinal cord. We then get to posteriorly the cerebellum. The cerebellum is going to be responsible for motor coordination and innate memory recall. That is, recall that we don't have to cognitively process out. It's kind of a rote thing. Once it happens that there's neurons and axons that are being projected from the cerebral cortex down through what's referred to as the arbor vitae. Notice the white branched structure going through the cerebellum, out into the gray matter, which is where we see the Purkinje cells. In those areas where we have the foles and the vermes are the ridges, the foles are the invaginations. The vermes come from the Latin term for worm, and they actually do look like worms. Hopefully we'll be able to take a look at them at some point in time this semester on one of the cerebral cortices in the cadaver room. The cerebellum will have projections known as the cerebellar peduncles, which will head superiorly into the cerebral cortex, as well as cross-communicate with the pons, known as just anterior to the fourth ventricle, and into the medulla oblongata. The pons, the medulla oblongata, have a bunch of motor tracts, as well as a bunch of sensory tracts that are coming in and out of them. They were referred to as the nuclei of the pons and the medulla oblongata. While the cerebellum is responsible for innate information recall and motor coordination, the pons and the medulla oblongata are responsible for cranial nerve control of facial and cervical motion, as well as regulation of autonomic responses. These are the areas that are going to be involved with the autonomic reflexes. The book will say that there's a cardiac control region. There's no real cardiac control because the heart does what the heart wants to do in terms of its regulation. However, there are some cells coming out of there that's going to adapt the heart in terms of its function based off of sympathetic and parasympathetic signals. So if we continue on and look at the pons and the medulla oblongata, you can look at it both in cross-section as well as in terms of a frontal view. Notice there is another part of this structure that we didn't talk about before. That's the reticular formation. The reticular formation is going to be responsible for activation, which is why it's also called the reticular activating system. And what it's responsible for is it's responsible for kind of turning the cerebral cortex on and off and getting the body ready to do what the body needs to do. So it's going to activate the body. There are a couple of structures that we have to be keen on because they're going to be involved with a lot of responses that we're going to look at with special senses. We also have to look at some of the nuclei for most of the cranial nerves are going to be coming from this area. In particular within the pontine nuclei, as well as the olive nuclei. In between the olive nuclei and where we see the reticular formation, we have spinal tracts, motor tracts, which we refer to as being the extrapyramidal tracts. Those are going to be responsible for movements outside of normal somatosensory cortex signals to cause movements. This is where we get the parallel and reverberating pathways coming into play with use of the cerebellum to make sure that what we want to do for motor is what we actually want to do for motor. I'll get more into this when we talk about the peripheral nervous system and how we get the motor signals that we want to get.